Norosoa Razafinarivo, Norosoa changed a little bit the title of her presentation. So the title will be Coffea Genome Size and Genetic Diversity using Nuclear Microsatellites Markers across Africa and Indian Ocean Islands.
So thank you. Good morning, ladies and gentlemen. So I'm pleased to present my work entitled Coffea Genome Size and Genetic Diversity using Nuclear Microsatellite Markers across Africa and the Indian Ocean Island. This work is the result of a collaboration between FOFIFA in Madagascar, IRD, Nestlé and INRA in France, and the Kew, the Royal Botanical Garden in United Kingdom. So, the Coffea genus belongs to the Rubiaceae family, the fourth largest flowering plant family. The Coffea genus, including X. Psilanthus genus and X. Baracoffea subgenus, includes 124 species, and there is a free secondary center of diversification of Coffea, West and West Central Africa with 24 species, East Africa with 22 species, and the main secondary center of diversification is Madagascar with 61 species, namely Mascarocoffea. So, what do you know about genetic relationships between species? Genetic relationships between species was assessed using SSR markers in 15 populations. This study relieved geographical patterns and confirmed the existence of 4 main geographical regions: West and West Central Africa, Madagascar, East Africa, and Mascaran. The genome-wide polymorphism pattern using REMAP and SSAP results Coffea species lineage. But there is a few data for Malagasy species and known data for the Comorian and the Mascaran species. At the cytogenetic levels, the genus is very simple. All Coffea are diploids except Coffea arabica. The genome size for African species was first analyzed, and there is a broad genome size range from 1.03 to 1.76 picogram for 2C, and the general trend is to increase from East Africa to West and Central Africa. The chromosome organization was studied by using fluorescent in situ hybridization of 5S and 18S rDNA-LOC, and there is 1 pair of 18S DNA-LOC and 2 pairs for 5S rDNA-LOC for West and West Central Africa. For East Africa, there is 2 pairs of 18S rDNA-LOC and 1 pair of 5S DNA-LOC. But there is 1 data for Mascarocoffea. So it was to assess the genetic diversity across and among the main center of Coffea species diversification and to estimate the genome size across the Indian Ocean Island and to compare the range of variation to previous data for African species. So let's talk about the genetic diversity across Africa and the Indian Ocean Island species. Here is the representation of geographical distribution of the species used on this study. So we used 26 species from Africa and 35 species from Madagascar and Mascaran Island. The genetic diversity was assessed using 13 nuclear microsatellite markers, and in this figure, you see the total number of alleles for Africa, for West and Central Africa, in blue. So there is a decreasing of allelic richness from West Africa to Madagascar, East Africa, and Mascarenhas. And the allele richness for West and West Central Africa is due to an important contribution of Coffea canephora. In blue, we can see the numerous shared alleles in each region, and in red, the private allele for each region. So the numerous shared allele increasing from West and West Central Africa, in Madagascar, East Africa, and in Mascarenhas, while private allele decrease from West Africa, Madagascar, East Africa, and the Mascarenhas. For genetic parameters all round, and there is a very high diversity of the size of leaves. There is a morphological diversity of young leaves. The color of young leaves could be red or brownish. Coffea flowers are unifloral or plurifloral. And there is the diversity of the color, size of the fruit. So the genetic diversity is associated to a high morphologic diversity in Madagascar, but the trunks deforestation and the political problem, the poverty in Madagascar and the lack of funds, thanks to the UCC for their help until end of 2012. And there is a high number of species have disappeared or are endangered. Kianzavat is home of a unique collection Mascarocophea over the world, and Kianzavat research stations needs your help to ensure the protection of this invaluable genetic resource. So I'd like to thank all FOFIFA authorities to facilitate our fieldwork in Madagascar. Thank you very much for your attention.
Thank you, Norsu. I think we have a very short time for a very short question and a very short answer. Microphone, please.
Thank you very much for your talk and the picture showing all this variability in coffee from Madagascar. You didn't show any gene flow analysis in Madagascar in your results, in your population genetic. In Madagascar between the populations that are still in the wild.
Kind of analysis.
Thank you. I would like, before ending this session, I just would like to continue on the appeal that Norsu started about the Kianzavat collection. As you saw, Madagascar is the cradle of a huge diversity for the Coffea genus. It's even the biggest spot where you can find the diversity of Coffea species. It's not only the case for Coffea. Madagascar is known as being a hotspot of diversity for many plants or animals. And concerning the, let's say, possibility to save this station, because it's really a question of life and death, this station harbors a marvelous germplasm bank which needs to be preserved for research, because some of the plants are now present only in this station, not in the wild. They disappeared from the wild. And an initiative has been initiated, and you will find a brochure describing, let's say, the situation of this Kenjerat station. Of course, I'm here appealing mainly to donors, maybe private companies or rich people present in this assembly. So I encourage you to have a look at this brochure, which will be at the registration desk, and maybe diffuse it among your, let's say, relatives and friends and anyone who can be sensible to the preservation of the genetic diversity of Coffea species. So now we end this session. We can have a final applause to all the speakers of this session.